BT8009 is a Nectin-4 targeted drug with MMAE as the payload, but actually it's a small molecule conjugate. It has a different structure to the ADC. It's called the bicycle because apparently it looks like a bicycle, though I think it looks more like a steering wheel from an arcade game to me. But if anyone else can show me where that, or even a hose pipe, but not a bicycle. But that's where we are with this. Single agent response rates, this is Johan Lorio's data, Juvelo, phase 1. Single agent response rates of 45%, EV was 40%. The adverse event profile looks quite attractive, actually. There's issues around whether there's less skin reaction and less neuropathy. It's only 45 patients. I think we need to wait and see. As I said before, cross-trial comparisons of adverse events of phase 1, very, very risky to do. This is also in a randomized phase 3 study. Again, it's very similar to EV302, GemCis versus this. It's also got a relatively complicated phase 2 program. Who would like to take on this drug and the future of this drug and where it goes?
I think that I will quote what Andrea mentioned before, that it's a matter sometimes of toxicity. When we envision a future, when we have more than
Do we need this drug? We've already got EV Pembro, we've got EV.
Yes, we always need more drugs because this is a novel mechanism, and I think we can build on this. Maybe not with this target, but I think this is a really interesting mechanism.
And also, we're going to see whether there is cross-resistance with EV. I think the company is looking into EV-resistant patients to see whether this has activity. I think we're going to end up in a future state, Tom, like RCC first line, right? We have 4 standard of care options there. Right now, we have that trial, phase 3, looking at this compound plus checkpoint inhibition versus the Javelin Bladder 100 regimen, and we have the DV plus Pembro versus the Javelin regimen. So we may end up in the future with 3 positive trials, and then how do you decide? Are we going to be forced to use biomarkers like HER2 or others? Are we going to look at toxicity or both to make a decision for
Carla,
patients?
what's your take on it?
So, I mean, I think that the one aspect about this drug that might be particularly interesting is the lack of neuropathy or lower neuropathy than we see with EV. That could be important. It might also be interesting that if EV Pembro moves earlier into the MIBC setting,
There's 16% grade 2 neuropathy.
Yeah, I mean, it's a little bit better,
50.
I think.
It's not 50, but it's still neuropathy. And grade 2 neuropathy that's not running around.
Right.
Neuropathy, CTC criteria are not great. Grade 2 neuropathy is actually quite serious. And so, but don't get me wrong, it does look, in indirect comparisons, it looks lower than EV.
A little better.
But it depends on how you grade them at home.
Yeah. And it is a different mechanism of action, which I think is going to be interesting, and we'll see how that plays out.
So it definitely has a future. So let's go for scoring a 10 out of 10. Because you might say, well, do we need another one of these drugs? It's very similar to EV. Assuming it's active, and assuming the randomized phase 3 is positive, how out of 10, how relevant is this?
I think it depends on which space you would want to use this in. If the future is EV Pembro moving in the neoadjuvant space, we do need such drugs.
Can I be a bit cynical and interrupt you?
Sure.
Very unusual.
Yeah. So look, EV302 comes in with hazard ratios of 0.4. Repeating that's going to be extremely difficult. There's more access to subsequent therapy. The trial conduct's going to be more difficult because patients won't want to be in the control arm. They'll be piling off looking for other things. So repeating those is going to be hard. So inevitably, this is likely to come in the 0.6s, not the 0.4s. If the results are if they're not as good as and GemCis Nivo was not, was in the early 0.7s and late 0.78 for OS. Do you have to have as good a result as EV Pembro to compete with it, or does it just need to be a positive trial? Because we've got positive trials with GemCis Nivo. The reason why EV Pembro has superseded GemCis Nivo was so positive. But if the results come in and they're not so great, is it competitive?
I think it would be in terms of the toxicity profile being different. It's good to, like Petros said, having multiple options like the RCC field, because the trial of this versus EV Pembro is never going to happen.
Shilpa, I'm going to interrupt you again. So the pazopanib story, we did sunitinib versus pazopanib. We did those non-inferiority trials, and pazopanib came out as better tolerated, Pisces, better quality of life, but there was always this issue about whether it was a bit less active, and it never really took off. Being first and having better results is going to be hard to compete with, isn't it?
I think yes, but the more we see such drugs and better patient-reported outcomes, better toxicity profiles, I think it's going to be very good, because one approach cannot be right for all patients.
Someone else pushed back on me a bit, saying not being first with not quite such great results is going to be very difficult.
Think about that atezolizumab in metastatic urothelial carcinoma. It was the first checkpoint inhibitor approved.
Yes.
Right now, there's no approval in the US. I don't say this will happen, of course, with EV-Pembro, not at all. But my point is that...
But 211 was negative. The randomized phase 3 with 10, whereas EV-Pembro came with a hazard ratio of 0.47 OS.
And I predict that EV-Pembro will remain the standard of care for a long, long time. It's very high bar to beat. It's very high bar. My point is, being the first there doesn't mean you're the only one there. So we have seen that in other tumor types. So I agree with colleagues that we need to have options. And at the end of the day, we're going to look at all these trials. I don't think we can compare hazard ratios across trials because exactly what you said, Tom. The subsequent therapies may be different, and that can impact the hazard ratio to multiple dose trials. So positive trial will be good. EV-Pembro is a very high bar to beat, but those trials do not compete against EV-Pembro.
But to your point, Tom, the comparison of the control arm, it's a bit too late, so that's why the future of this agent might not be in the frontline setting, but subsequent setting, post-EV-Pembro.
But we don't know there's resistance yet. We don't know that we can't give it post-EV. I mean, you would think that maybe you can't because it's the same payload, but we don't know yet. And I think understanding these molecules and attaching different payloads to them, my dream would be multiple payloads because, you know, as a medical oncologist, we give combination therapy, so why not? That's why I like the DADIO study, because I think that if you can attack the tumor from multiple different directions, that's the way to go. But we need the chemist to work on that. But I think the molecule is very exciting.
We've got the last drug here that we're discussing today, EGFR HER3 bispecific, topoisomerase the payload, data out of China, relatively small numbers, 22 patients. And you can see here response rates of 40% again, 41%. A novel target, obviously, completely different from what we've talked about so far. We don't know enough about this drug. On the stage at ESMO, they announced 2 randomized trials that they were going to launch, randomized phase 3s. We didn't get too much of the detail of those, whether they were China only or global. Who wants to take this on?
Sure, I mean, I can comment. I think it's exciting just because it's a different target. I mean, I'm excited to see the EGFR, I'm excited to see the HER3, like it's doing something different, and that itself is exciting for me. I think the objective response rates, this was in all comers, but I think in patients who had only one line of treatment, it was even a little bit better than that. So I'd be curious to see where that goes.
Any more questions on this before we move on this drug here? Oh, we're excited about it, I guess, but we don't know enough. Is that fair?
Yeah.
We don't know enough.
It's early.
Small study.
Petros.
Small study, we need more data, I agree, but that's interesting. It's an interesting compound.
It's an interesting compound, yeah. It's
sure.
just
Very
very
dual
Relevant
bispecific.
targets.
So
the nuance actually in the presentation, they quoted that they had data in the second line, but actually if you look at the actual data, they had also data in the subsequent line. So I think it's relevant to look at the detail. The drug is active.
22 patients.